Hello. The goal of my presentation today is to establish that virulence genes in Vibrio cholerae allow for toxicity, high transmission, and antibiotic resistances. Cholera is a pathogen which causes diarrhea and vomiting, and this can make it fatal in some cases where proper medical care is not taken into account. This pathogen contains genetic components which increases toxicity, which are called virulence genes. There are many of these that are involved in cholera itself. However, the most important one that we'll talk about today is the O1 antigen, which is present. Um, the antigens O1 and O139 are the only ones which are able to cause epidemic level infections. No other antigen has been found to do this. However, there are many more. It can be seen in this table that the virulence genes present in samples collected from the same area even can differ. So as you can see here, the strains, these three which were collected on the same date and from the same place, even the serogroups differ a little bit. Most of the virulence genes do not that were tested in this case. But you can go down the list and see that these had some minor differences as well. They were collected from the same place, and then there were some minor differences in some of the other ones. I feel like it is worth noting that none of these samples do have the O1 or O139 antigen in this case. However, they were taken from India in a time when there was an outbreak occurring. And these were all environmental isolates taken from water around the areas of the outbreak. This graphic and this one here show that the distribution of the virulence genes in cholera is inconsistent even in the same area. So as we saw over there, there were some differences in serogroups, some minor differences in the actual genes that were present. And this one as well kind of shows the same thing. However, this establishes a case where toxigenic serogroups or antigens are present as well. So as you can see, even these that were taken from the same source and of the same serogroup, there are some differences in the presence of CTX and some of the other virulence genes that can be seen over here as well. CTX is often an important gene for toxicity. However, it is not imperative that these toxigenic strains have it, just because they have the ability to do that based on their serogroup itself. However, it would increase the ability of these ones up here to become more infectious on their own. Because cholera is a pathogen which exists in the water supplies. It generally lives in aquatic environments. It is very easy for this to transmit through water supplies, especially in places where water sanitation is not of high importance. So often, since these non-O1 and non-O139 strains exist, they can also cause minor infections. So in cases of small groups of cholera cases, it could be assumed that these come from these which are just more toxic. However, we can see in this map that as you move further away from this large water source, the number of cases seem to increase in this area in Ghana. It is worth noting that these cases were generally pulled from hospitals, but it seems that there are more cases in the hospital that is further from this large water supply, which could mean that the water supplies that are over there are just more contaminated in this case due to the fact that they are further away from a large water supply and they likely do not have effective water sanitation methods. That being said, as cholera moves into human populations, it can be treated with antibiotics. There are some issues with this, though, given that cholera has developed antibiotic resistances over time. So we can see in this period of time from 2012 to 2015, there was an outbreak going on in this area, and the antibiotic resistances are changing fairly steadily over time. You can see the introduction of new ones that didn't exist in 2012, whereas in 2014 they were most of the strains were resistant to erythromycin in this case, and then that kind of decreased in 2015. So this can be partially attributed to virulence genes and the fact that they are changing because they do have some amount of horizontal transfer between them, between different strains of the pathogen. So you can see how there are new introductions of antibiotic resistances over time, and based on which ones they're being treated with. It is also worth mentioning that the non-outbreak isolates do have some of the same resistances, but often these are pretty different from what we see above. In this following study, isolates were taken from Lake Erie and its adjoining river in this case, and you can see all the sampling sites here. And all of these samples were taken and they were genetically sequenced and tested, and only 2 actually, you can see down here, were positive for the O1 antigen, and none were positive for the O139 antigen. Now, in this study, the transmission wasn't explicitly determined. There was not a survey of cases in the surrounding area. However, based on the fact that there are only 2 isolates from this sample that were positive as O1 serogroups, and none that were O139, and the fact that this was a sample taken from the United States, it would be safe to assume that there were not many cases in this area due to the water sanitation practices and the fact that there are low amounts of toxigenic strains. This is a very low percentage in this case. But we can't obviously give explicit data on that because it does not exist in this study. So we can look through this graphic and notice that there are some differences between the different virulence genes that were tested in this case. I'll move this over to this side so we can see that differences in which gene was being tested in this case. So even between these 2, which were taken from the same area, there is differences between the proteins, the different genes, and so on. And this continues down the line. And then in this next graph, these isolates were taken and they were tested for antibiotic resistances. In this case, S means that it was susceptible to the antibiotic, R means it was resistant to the antibiotic, and I means there was intermediate resistance to the antibiotic. So even as you can see here, isolates taken from the same area did have different resistances to erythromycin in this case, nalidixic acid, and streptomycin. And then this continues down the line. You can see that the differences in the antibiotic resistances occurred even from samples taken from the same area. And then I feel that it is worth noting that these 2 samples, which were positive for the O1 serogroup, were the only ones that were fully resistant to streptomycin in this case. It can almost be thought that due to the fact that virulence genes do change in cholera, due to the horizontal transferring, that this could be the cause of why some of these antibiotic resistances are changing. So it has been shown that virulence genes in cholera do allow for toxicity, high transmission rates, and antibiotic resistances. Thank you.